Okay, I just want to thank Topcon for inviting me to give this discussion on the evolution of OCT imaging, Swept Source OCT. So just a little background, I've had the opportunity to work with the Triton device since about 2014 now. And over the last several years, we've had a lot of experience in using it in routine day-to-day clinic with our regular, straightforward patients, as well as sort of in the more complex space, the surgical space, and even in the wide field OCT space. So there's a lot of utilities for this product, and I think, pictures are worth 1,000 words in some of these cases. So hopefully this will be instructive and helpful as you head out and discuss it with other potential OCT users. So the evolution of OCT, OCT has really taken off in the last several decades. We've done an amazing job at pushing this technology forward in ophthalmology. We started off in time domain OCT, which you can see on the far left. And many offices and practitioners are still living, obviously, in the spectral domain space, the 840, 850 nanometers. And, the Triton, of course, lives now in the 1050 nanometer world with Swept Source, and there are some other prototype devices that have slightly longer wavelengths. And we're going to sort of see how the two technologies, spectral domain and Swept Source, stack up against one another, and also where the utility for Swept Source is in routine practice. So as I mentioned, obviously we've undergone a major revolution over the years in terms of a variety of devices that have come to market. And the goal really is, better, faster, and hopefully cheaper, but that really hasn't been realized, of course, because these technologies are quite expensive. But they do come with significant advantages. So we started off with the OCT-2000, made by Zeiss in '96, and then the Stratus sort of became the most commercially and commonly used OCT in clinical practice in 2002. And then over the years, we've taken some major leaps forward. Topcon has introduced several devices, including its 3D OCT, obviously the Maestro. And then we moved into the prototype for the precursor for the Triton, which was known as the DRI OCT Atlantis. And actually, I had the opportunity to work with that and give some feedback to the team back in Japan. And it was also a very, very revolutionary device. But obviously, the Triton has really landed in the right space from a balance of technology and clinical utility. And of course, there is still another competing Swept Source platform, the Zeiss Plex Elite 9000, which lives largely in the research world. So we've seen some big leaps come up in terms of the years as they've gone by, and our devices have gotten faster. We're now in the 100,000 A scans per second, which really translates to being able to acquire large amounts of data very, very quickly. So briefly about spectral domain, I think, some of the science is not so relevant for day-to-day sales purposes, but it is important to understand sort of where the technology does come from and the origins of it. Spectral domain OCT relies on a broadband light source with a reference mirror and a spectrometer, which then basically allows reflected light that comes back from the retina to be captured on a reference mirror. If we took a look at Swept Source OCT, the process is also still very similar, except in this case, the Swept Source laser head is a tunable laser and operates at a higher wavelength of 1050 nanometers. So what does this mean in terms of comparing the two technologies? On the left, what you can see is a graph that compares the sensitivity versus the depth of the imaging. So the goal here is obviously we want to try to get as deep as images as possible of the eye, but not lose sensitivity. So you can see with the red line, the spectral domain line, as we try to penetrate more deeply, we get sensitivity roll-off, it's called. Whereas on the green line, you can see the Swept Source OCT, because it has these longer wavelength, we're able to achieve higher sensitivities at greater depths within the eye. So when you look at sort of these two approaches side by side, obviously the wavelengths are different. And the A scan per second is 100,000 for Swept Source versus spectral domain, which is 70,000. And again, that makes basically this new technology, the Triton, 5 to 10 times faster than spectral domain. And that really does translate to a faster throughput in the office. And we'll talk about how to use that element when discussing this product in the sales space. And then, of course, as the graph shows, there's better sensitivity with Swept Source than spectral domain and an improved signal to noise ratio. So let's take a look at how it looks like in clinical practice. Here is, one of the gold standard OCT devices that many of us have used in the past, the Heidelberg Spectralis, probably one of the better spectral domain devices that's out there that's not a Topcon unit, of course. And you can see here the zero delay line, which is visible in green. And as we try to image deeper and deeper into the eye, you can appreciate that the vitreous space, which is in black, becomes more cloudy with noise, and we're losing sensitivity. And if you look at the architecture of the retina, you also start to lose information of the choroid, and also some of the structure of the retina becomes less defined. So we'll take it back a notch again. Here's where we started, right? With our zero delay line, everything looks nice and crisp and sharp. And as we try to get deeper penetration, what ends up happening is we get a sensitivity roll off. And so this basically is due to the fact that the 850 nanometer wavelength has a significant drop off in sensitivity with deeper attempted penetration. So here's the two technologies side by side in the same eye. This is one of the commercial images that's commonly shown for the Triton. And you can see the same phenomenon, that as we really get deeper and deeper with the bottom images, the images themselves become grayer and less defined and less clear. So the sensitivity drop off that happens with spectral domain is really what is a limiting factor with this technology, and it's ultimately where Swept Source fills in the gap. So one of the other beauties of the Triton that we've seen, and of Swept Source in general, but particularly of this device, is that because the wavelength is longer in the 1050 nanometer realm, it's actually in the invisible world of light. So it's an invisible scan. That is, the patients do not see the light that is being used to scan their retina. Whereas with the 840, 850 nanometer laser, they're in the visible spectrum of the light spectrum, and as a result, they do see a flashing light. So one of the advantages of imaging patients today with the Triton is that the patients are much more comfortable, they're much more stable, and obviously those two facets, a comfortable and stable patient, allows for much more better imaging and also faster imaging. So here's, we have the fortune of having multiple OCT devices in our office. We have the OptiView, AngioView, we have the AngioPlex from Zeiss, we have the Plex Elite, we have the Triton, and we're fortunate to be sort of a think tank center for OCT imaging. And we actually oftentimes get the opportunity to do some head-to-head work, and this is an example of how we see things with respect to OCT angiography, as well as just the interfaces. And every device has its own pros and cons when it comes to interface, but on the left, you'll see here the OptiView AngioView, which is a good device. And on the right, you'll see, of course, the Triton. You'll see sort of the amount of time that it takes to capture the same patient's images and also do an OCT angiogram on those patients is quite a bit different. So I'll let the videos run here. You can see on the far left that we're starting the scan for this patient's OCT-A, and we're starting the scan on the far right for the patient's OCT-A on the Triton. And one of the factors that's sort of significant here is obviously having a touch screen, which is a nice added bonus for the technician. Our technicians ironically fight over which person gets to use the Triton on a regular basis, which I think probably speaks to how they've made it very friendly for the user, which obviously is not the physician on a regular basis. And, in terms of the tracking element of keeping tabs on the eye while the patient's eye moves, having real-time tracking for the motion correction in OCT angiography is a bit different than in the case of the OptiView. And so what you can see here is that, the OptiView uses motion correction technology, and as a result of the motion correction technology, it's a lot slower. So OCT-A, I know at the moment, is not presently FDA approved, but again, it involves a series of B scans that are captured and then brought together in order to evaluate blood flow within the eye. And of course, every common OCT device now has integrated OCT angiography. And it's a bit labor intensive from the practice side to be able to get good, reproducible, high quality images. And that said, it's very important to have, once again, a stable, cooperative patient that can be imaged very quickly so we can limit the effects of motion. So here's an example of a fluorescein angiogram, and you can see the non-perfusion in this patient that has a branch retinal vein occlusion. All the dark patches represent non-perfusion, and the OCT angiogram from the Triton shows the same thing. So the question really is going to become, do we still need fluorescein angiography? And in our practice, we are constantly asking this question and trying to push the envelope forward to have less invasive testing for our patients that is safer, like OCT-A, and see if that can obviate the need for using a fluorescein angiogram. So very briefly on OCT angiography, You can see here that these are all the normal layers that we commonly see with respect to the vasculature of the eye. There's a superficial capillary plexus, there's a deep plexus, and there's a choriocapillaris. These are all images from the Triton, which does an amazing job at providing high-level detail with OCT angiography. And so here's another example of a normal eye that we would commonly see with the interface of the software. And again, the software is designed to give us a view of all slabs simultaneously with the ability to scrub or navigate and adjust segmentation to identify pathology. So it's actually a very easy to use software that gives you everything you need in a single one-up type image. And once again, this sort of shows that same comparison. We look below, we have a Spectral-Domain OCT device, and on top we have the Triton. And one of the notable differences is going to be seen in the blue boxes. So in the blue far bottom right box, you'll see some vessels from the superficial capillary plexus that are projecting onto the choriocapillaris. And that artifact is called projection artifact. And so the Triton has the ability to remove projection artifact, and I think many of the devices are now shooting towards this effort of making the images clean and free from projection. And you can see that there with the yellow arrows. The optic nerve is a very hot area to image with respect to OCT angiography. In fact, I think one of the marketing approaches should be to look at getting this into the hands of glaucoma specialists, because it's been shown that the teal box that you can see here, the radial peripapillary capillaries, are the first to drop out when we have a person with glaucoma, and it can be the earliest sign that things are afoot with respect to possible glaucoma, which can therefore lead to nerve fiber layer loss. So it's not a tool that we should be just focusing on getting in the hands of retina specialists, but also in the hands of glaucoma specialists, because obviously, as we know, optic nerve damage is irreversible. So similar to the spectral domain device, this does a nice job of giving us all that information, but of course the Triton does this very quickly, much more quickly than spectral domain. One of the other small differences you can see here is some of the distortion that can be induced as a result of, taking an image in the spectral domain, whereas with respect to the Swept source, we don't see that slight distortion. So here's an example of some of the segmentation software. Obviously, we were able to look at the choroidal neovascular membranes very clearly, which you can see here. This is one of my patients that has wet macular degeneration, that sort of Medusa head looking structure in the green box, teal box, and that appears green in the depth decoded image is the choroidal neovascular membrane. And of course, we can do real-time videos with our on-FOSS imaging to sort of better highlight this, and it's a nice way to do some teaching for your clients so they can appreciate the fact that they themselves can control the extent to which they evaluate the lesion. Here's some nice images by Carl Glittenberg. Obviously, he's been a great addition to Topcon, and you can see sort of another view of what otherwise would be considered a disciform scar, but in fact is a CNVM, and it's active in the case it has some fluid surrounding it. And another image from Carl, once again, sort of showing the same thing, but a larger CNVM. And again here in a patient with what looks like wet AMD. So how is this technology useful? Well, basically where we're going with OCT angiography is being able to track the size changes over time. And so what we'd like to be able to do as physicians is sort of see the growth of these lesions over time or shrinkage in response to therapy. And that's basically what we're seeing sort of with some of our clinical trials that we're doing with industry. And that's really who mostly cares about this, to see the response of these vessels in response to their particular therapeutic agent. So here's a publication from Dr. Saraf out of UCLA that's basically showed anti-VEGF treatment resulting in shrinkage of CNVM. So, as the OCT-A piece of it becomes FDA approved, this will be a value add to the Triton. I realize right now this discussion is largely off-label, but for the purposes of showing the full potential of the Triton, I think that this discussion is still valuable. And of course, without good speed, you can't get good imaging. So you can see here on the right, this is a volume rendered image, and Carl's helped put this together for me. And, with having a lot of data acquired very quickly, you can get, three-dimensional volume rendered images, like in the case here of this branch retinal vein occlusion. And you can appreciate sort of elevation of the retina and the fluid and all the vascular detail. And this kind of information cannot really be gathered as quickly and as readily with spectral domain. So let's talk about, everyday usage for the Triton. I mean, I think one of the advantages really is in the fact that the wavelength being 1050 nanometers is able to penetrate through a lot of bad media opacity. So on the left, you'll see a patient that has had macular hole surgery post-op day 1. That's the OCT. And you can actually see the fovea very clearly. You can see that the hole is largely closed. And you can see the color photograph from the Triton itself. And one of the advantages, again, is that the Triton does color photography, so you can have a registered OCT to a colored fundus image. So you can confirm that what you saw clinically is actually what you're seeing on the OCT. So this patient had 95% gas bubble in the eye, and we were able to image through the bubble in its entirety. And we actually published a series of 35 eyes with macular hole surgery on post-op day 1 and post-op day 3, even showing excellent penetration and capturing of the macular hole status. So gas, no problem. Looking at cataract in the middle, obviously the aging population cataracts are very common. And having the Triton image through cataracts, even the densest of cataracts, can give you an excellent view of the retina. You can see this patient has wet AMD with a CNVM and fluid on the OCT, and the cataract itself obviously is like a 3 to 4 plus cataract. On the far right, is one of the other surprises of this device is that it's able to penetrate through blood very easily. The image below, the color photograph is from the Topcon DRI-50DX, and it shows a lot of pre-retinal hemorrhage. There was no view of the macula. We did our Triton OCT, and you can clearly see that the macula is actually normal. Above the macula is blood and neovascular tissue under the hyaloid of the vitreous, as well as on top of the hyaloid. So the patient eventually actually had surgery and did very well. But if we did not know the macular status, this would really make our conversation with the patient a lot more tenuous. So we talked about the fact that the light is in the invisible spectrum. And so people, obviously as we get older, our pupils tend to become less reactive. And you can see here a very common patient that we see in our office, someone who's got a small pupil. And even with a small pupil, because the pupil is not responsive to any light with respect to the Triton, that is, the Triton doesn't stimulate the pupil because it's in the invisible spectrum, we get a very clear view of the retina even through a small pupil. You can see the patient has an ERM with some drusen, and as a result, you can also see that the infrared image, which is all blurred out, of course, because we don't get a great infrared image through a small pupil, but we are able to get an excellent OCT image in this case. So, nice example of how we can use this in an undilated pupil. And I can tell you in our practice, we went from doing our injection clinic with patients getting dilated, then getting OCT'd, and then getting injected because they were having a spectral domain OCT, to not getting dilated, which saved us 15 minutes per patient at about 35, 40 patients per session. And I'll let you add that up. And that really added to a considerable amount of time per patient. And patients really remarked to us how much more comfortable the imaging was and how much faster their visit was. And so we've adopted this practice now since about 2014, and we've been really successful in getting all the results that we need. Of course, there's always going to be 1 or 2 people every so often that do need to get dilated because they need to have that extra access. But for the most part, 99% of our patients do not need dilation for OCT. So I talked about gas a little bit earlier. So this is an example of a high myopic patient of mine that had macular hole surgery, and the OCTs on the far left of the screen are the OCTs that are done through gas. And you can actually appreciate the fact that you can see retina, you can see sclera, you can even see orbital bone, which is quite impressive. And then you can see here this meniscus of gas at the macula right in the center, bridging the 2 areas of the macular hole, which is yet to fully close. So, from a surgical perspective, Swept Source OCT has been a game changer. It allows us to get through gas, blood, oil, cataract. All these features are significant in clinical practice, specifically for the surgical physician. Here's another example of a patient with a bad anterior subcapsular cataract, and you can see up top is the image from the anterior segment lens, and you can appreciate that the cataract is densest sort of towards the anterior or the iris plane. And the infrared image is obviously in the middle, and then you can see obviously the retina looks clear. So, no matter sort of how the eye tries to impede the Triton seems to prevail. So really, really nice for the physician and ultimately benefiting the patient's care. Here's a head-to-head example on the far left. We published this in Ophthalmology last year. It's that case I showed you with the pre-retinal hemorrhage on the left in figure A versus the spectral domain equivalent of a pre-retinal hemorrhage in figure C. And on the bottom left in figure B, you'll see the Triton image, which gets right through blood and shows you all the retinal architecture. And on the bottom right, you can see the spectral domain image from Heidelberg, where the blood is causing backscattering and you do not get any retinal detail at all. So again, highlighting the beauty of a long wavelength penetrating blood and media opacity. Sometimes we have opacities in the vitreous itself. On the right, you can see this sort of snowstorm that sometimes commonly presents itself in a patient with a condition called asteroid hyalosis. Clinically, it's hard to see the retina at the microscope. It's hard to see the retina unless you do a fluorescein angiogram. And so OCT itself is really the only thing we can rely on. And with Swept Source, these small calcified parts of the vitreous do not pose a threat, and we can get a very great image of the retina, as you can see on the left. So this is an example of Plex leaked, just for comparison purposes. It does show you that this is a Swept Source device, which provides pretty good imaging also, and this will be a topic of conversation when it comes up when you're on the sales pitch and talking to your clients. So it is important to be able to realize what these images do look like. And we get the same exact images from the Triton. So from the perspective of, looking at the images themselves, they're really, really equivalent at this stage, and there's no real advantage to having a Plex type device. And then also, from the perspective of what the device does, this device just does OCT, whereas the Triton does, the full perspective of retinal treatment, or from color photograph to fundus autofluorescence to fluorescein to OCT, on-phos OCT, and OCT-A, as well as anterior segment OCT. So, I think when you look at devices head-to-head that are in a similar class, that is Swept Source, it's important to recognize the major differences. And really, the Triton is an all-rounder that does everything that a glaucoma and/or retina and/or generalist or optometrist would potentially want in their practice, versus a research-only device, which has just one capability. So when we talk about techniques of imaging, one of the other beauties of the Triton is that it has an internal and an external fixation light. And these small features, which are seemingly not exciting off the top, are important because, at least in our practice, we pay a lot of attention to wide field imaging, which really involves imaging outside the posterior pole. And we've actually developed techniques with the Triton to allow patients to be imaged in a wide field format. So this is from one of the papers that we published, and basically shows that you can use the internal and external fixation light, as you can see with the bottom left and bottom right images, to really image way out to the periphery. So some of the images that I'm going to show you now are all from the Triton, and they're all Swept Source wide field images that are achieved by using the internal and external fixation lights. And not all the OCT devices offer the duality of this type of lighting. So having this as another feature can assist in marketing the device in the context of wide field OCT. As you can see here, we were able to montage several OCTs together to get about 117 degrees of arc length in this case. So here are some examples of how we can get great Swept Source imaging of unusual pathology. This is a patient with an astrocytic Hamartoma, which can sometimes be linked to a brain lesion. This was a child that I saw in my practice, and the OCT features of this lesion actually are linked to a brain lesion, which he did have, and it was dealt with very early. So, wide field imaging has a purpose beyond just cool pictures. Sometimes it can actually be life-saving. And there's the OCT-A of the same patient using the Triton, which actually is also possible outside the posterior pole. And we look at sort of very far-reaching lesions. This is obviously an optos fluorescein angiogram of a capillary hemangioma. And below, you can see very nicely that, we are way out there at about 100+ degrees, and we can get great imaging of the lesion out in the periphery. And this was also done by, navigating the patient using an internal external fixation light. As I mentioned earlier, surgical applications are vast. As surgeons in retina, we do put in gas bubbles, oil bubbles, etc. And when we do that, we usually limit ourselves in terms of being able to image the retina. Here you can see a patient that had a pneumatic retinopexy and still has some fluid both on the radial and vertical scans through the gas. So, gas, again, not an impediment, and another advantage to sort of getting this in the hands of the surgical, retinal surgical person. High myopia can be a very difficult thing to image, but you can see here the Triton does a nice job of that. One of the other beauties of the Triton is that it has long line scans. And so what that means is that traditionally we've looked at line scans that are 3 millimeters, 5 millimeters, 7 millimeters. We now have 9, 13 millimeters, 16 millimeter line scans. And so the longer the line scan you get, the more real estate of the retina you can image. And if you can montage those together, you can get an even wider image. So, I think having long line scans is another advantage of the Triton. And again, it is able to acquire this image very, very quickly. Here's an example of a patient with Myopic Foveoschisis. And something that we see very nicely with Swept Source is vitreous detail. So you can see here the vitreous very clearly. You can see the schisis cavities and the separation within the retina. And all of that really allows some surgical advantages for planning for surgeons when they're looking into doing surgery. So this device really allows us to set ourselves apart in the practice by being able to see things that other offices just can't do. Here's an example of a macular hole. This is a very wide field image that was stitched together by our photographer, and you can see the flap of retina connected to vitreous way out into the periphery near the vortex vein. So, a nice example of how you can sometimes really MacGyver the situation to get some excellent imaging, but you wouldn't be able to do this with every single device because the data acquisition can be difficult and the clarity is not there. We talked about depth of penetration. So here on the left, you can see a deep choroidal lesion called the choroidal granuloma, and it's very clearly visible. And you can appreciate the fact that, we've got a wide line scan that's going on here, but you can get a great detail of the choroidal pathology. So, as we wrap up, I think, ultimately, Swept Source OCT is really a big game changer. It allows higher sensitivity. The patient stability is much better. They're much more comfortable. Of course, you get deeper penetration, all of which lend themselves to excellent technology in ophthalmology. The penetration is fantastic through media opacities like blood, oil, cataract, gas, as I showed in the images. The view of the vitreous to the choroid is pretty much synchronous, and you get to see everything in one motion without adding features like enhanced depth imaging, which we see with other spectral domain devices to help improve choroidal visualization. You can do that very easily with just the Triton. The fact that the acquisition of the speed of acquisition is faster really allows for a less motion effect when it comes to your patients. And ultimately, you look at the growing applications of Swept Source OCT for macular and peripheral disease. Those are expanding. And then, really superimpose on OCT angiography, which is super motion sensitive. And a device that's able to capture high level detail very quickly will ultimately win the OCT battle. So I think, this is an excellent device. And really, when you think about where we're headed, this is actually another example. This is the same conclusions, but just to further highlight the fact that we can get great information. This is a patient of mine with a choroidal neovascular membrane, and I work with Carl to sort of render this together so we could do a fly-through of her blood vessels. And you can see, it's really a significant wow factor for us to look at disease in a way we've never looked at it before, which is really from the inside out. So, this is just something we have not done before. We're on uncharted waters, and it was only really possible through a device like Triton and Topcon's forward thinking.